we were learning about last time. So last time we started talking about blood vessels. We're going to continue that this time. We're going to get through our tour about blood vessels, and then we're going to talk about what we call hemodynamics. So a little bit of physics, if you want to consider it that way. Talking about blood pressure and blood flow and exchange and all of those important things that blood is going to have or do, okay? So what are a couple important things that we learned last time in terms of blood vessels? Okay, three major types. So we have types of blood vessels. So we have three major types. We have arteries, we have capillaries, and we have veins, right? Arteries going away from the heart, capillaries exchanging materials with the tissues, and then veins returning the blood back to the heart. We also found, though, that there are kind of subcategories as well. So for instance, we have arterioles, which are small arteries, which also have the three layers that we were learning, but they have a particularly important role where they are working to dampen all of that pulsing of blood that we see in the arteries previous to that to protect the capillaries that come after them, okay? So they're helping to regulate flow, regulate pressure, and protect those capillaries. So we have arterioles as well, then we go down to capillaries, then we work our way up to venules, which are small veins, and then we go up to larger veins, which return that blood back to the heart, all the way to the other side of the loop. The last thing that we saw as well, though, is that there are even three types of capillaries. So we see a difference in structure and function even with these three types of capillaries. Continuous capillaries, which are very numerous, these are the most common ones. Fenestrated capillaries that have holes in the cells themselves, inside those epithelial cells, and then sinusoids, which have huge cells not only inside the epithelial cells, but also in that basement membrane. So we see some additional examples of blood vessels there. Okay, so definitely types of blood vessels are going to be important. Knowing their anatomy, do they have three layers or not? Do they have specialized anatomical structures which give rise to their particular functions? So we can see, like I said, multiple choice questions like that. We can see short answer questions related to that, things like that. All right, other things, another major thing that we talked about last time that was important or confusing. Or both. Yeah.
Wait, what are the three layers? You have the basement membrane.
So that's a good point. So when we talk about structure and function, most of the blood vessels have three layers. So when we're looking at their anatomy, right, and when we're looking at them in the microscope, let's say, when we're looking at large blood vessels, we have three layers to them. Three layers or tunics, right? We have an externa, we have a media, and then we have an interna. So that holds true for our largest blood vessels, arteries, arterioles, venules, and veins. All of those have three layers. But the last example that we were just having right there in terms of capillaries, they don't have those three layers. So what we find is that in capillaries, because their function is to allow exchange across the wall of the blood vessel, we want that wall to be as thin as possible. So effectively, doesn't really have a tunica externa, doesn't really have a tunica media, just has that thin epithelial border to it, or lining, and then it has a very thin basement membrane, and that's it. So it's a single layer of cells, flattened cells to boot, which makes it very, very thin, makes it easier for things to get across. And like you see with these different types of capillaries, if we need it easier still, we'll actually have holes in the cells or holes in the basement membrane, which facilitates the movement of larger molecules or even cells to be able to enter and exit the blood vessels. Okay? Venules, which come after the capillaries, are in fact a place where some of these cells can enter and exit also, mainly exit when we're talking about white blood cells. So white blood cells, remember 5 different types of white blood cells, that's important. 5 different types of white blood cells, each has a different way of protecting us. But for the most part, the ones that you see in your microscope slide, or we talk about circulating in the blood, those are just representatives of the entire population of white blood cells that are circulating throughout the body, outside of the blood vessels, like a security team on patrol within the tissues, etc., right? You can't protect the body unless you're out there amongst the people, amongst the cells themselves, right? So a lot of the white blood cells are outside of the cardiovascular system, and they will exit at the capillaries, but also at the venules. So in between the cells and places like that in these intracellular clefts. Okay, pretty good. So let us then, we're going to move this over to the PowerPoint, and we'll continue on from there. Okay, so regardless of the type of capillary, these capillaries, like you said, are most everywhere in the body. So they're going to be next to almost every cell in the body because that's where we're going to be exchanging. And when we say exchanging, to be sure, that is a trade. So make sure that you're remembering it's not just delivery. Think of it as the postman or something like that, or the postwoman. As they go into your neighborhood, right, they are delivering mail, but they're also going to pick up mail. So if they're only doing one part of the job, they're not doing their complete job. So these capillaries are going to be everywhere, and they're going to be exchanging with the tissues. Now, in truth, when we look at a capillary, okay, we're going to have cells in here, so we might have our red blood cells going through. We're going to have plasma in here as well. We said that in the plasma we've got tons of different molecules and ions and important things going through there, hormones, etc. Then we have our cells, a generic cell, out here. Now, in most all of the tissues of the body, we're also going to have what we know as interstitial fluid that is surrounding those cells. So what we have here is we have plasma, which is an extracellular fluid, right? Because it's outside of the white blood cells and the red blood cells, etc. We have interstitial fluid, which is an extracellular fluid because it's outside of cells. And then we have inside of cells, which is going to be an intracellular fluid, right? Within the cell fluid. So these are the three compartments that we're actually exchanging fluids, ions, and molecules across. So in truth, when we say that we're exchanging with the cells, if we get technical about it, there's actually a two-step process involved here. So the cells will exchange across their plasma membrane to the interstitial fluid, to the surrounding extracellular fluid in the tissue. Then those same molecules or ions or whatever will exchange across the wall of the capillary and get into and out of the blood compared to the tissue itself. Okay? So this is a two-step process of exchange. It has to go across this membrane here, the plasma membrane of the cell in the tissue, and it has to get across this wall here, these cell membranes of the wall of the capillary. Okay? So that's something we'll talk about. We have a chapter that's coming up a bit later. I think it's after respiratory system or something like that. It talks about fluid balance and pH and things like that in the body, which is pretty important for health science careers when you're talking about maintaining pH and maintaining fluid levels. That's a lot of what you're monitoring for your patients. So we'll get into these details a little bit further and talk about major ions and molecules found in these spaces, etc. But we said that for these capillaries, that's their main role, is to exchange with the tissues, and necessarily they have to have a thin wall to be able to do so. So they have to be numerous, they have to be small, and they have to be thin. And so oftentimes we have these things that we would call capillary beds in. It's not just one vessel going through this whole tissue. We've got to get in amongst all of those different cells. So we have a full capillary bed that's going to get in amongst the cells in that way. Okay? So generally, there can be like 10 to 100 capillaries per capillary bed. So it forms a very diverse network. So this is a figure that kind of shows that. And not only does it show our capillary bed here in the center, but it shows the arterial, right, the small artery leading up to it. It shows the venule, the small vein that's draining from it, but it also shows these branches that tend to route through it. So there's a couple interesting aspects to it. The meta-arterioles are actually leading into the capillary beds, like we see in through here, okay? So the meta-arterioles are kind of a transition between our arterial and the full capillary bed. But the other interesting thing is, you see these little bundles here, or even here in yellow? Those are called precapillary sphincters. And precapillary sphincters are bands of smooth muscle that will constrict or dilate the vessel leading into the capillary bed. So we said that it's important for us to be able to control blood flow and delivery to the different tissues and cells of the body, but it's based on what they need, right? So we said that we can shunt blood like to the stomach and to the small intestine if you've eaten a big meal like after Thanksgiving, or we can route it down to the lower limb if you're running or something like that, okay? So when we're doing that, we can certainly do that on the order of like muscular arteries and arterioles and stuff like that because they have smooth muscle. But even into these capillary beds, we don't have to run fluid into the whole floodplain, if you will, if that tissue isn't very active, okay? If it is active, yes, we need to get blood to all of these different cells, so we need to make sure that we let it go out throughout these different capillaries. But if it's not a very active tissue at the time, what we'll do is these precapillary sphincters will temporarily decrease or even cut off the flow to the full capillary bed and route it through this thoroughfare channel straight into the venule, okay? So what they can do is a bit like, particularly if you're like in Gilbert or something like that, where there's a lot of agriculture still, you know, they have waterways, canals that are running to the different fields. And so it'll be going along the edge of the field, but if there's nothing in the field, right, if they just harvested corn or something like that, and it's just dirt, why are we going to send water through there, right? So we leave it in the main thoroughfare channel, it can continue on. But if we planted, or if we're growing corn or something like that, then all those different valves along the way, we've got to make sure that all those are open so we can flood the field with the water and get that to the cells, in that case, that need it. So that's what these precapillary sphincters do. So we're always going to go into a capillary bed, but if it's not a very active tissue, we're going to route it right through this thoroughfare channel like this. And if it is an active tissue, then we'll open up all of those valves and we'll let it totally percolate or perfuse through all of the different capillary beds, okay? So there's a lot of activity that's going on down there, a lot of vaso or vessel activity. This is called vasomotion, where we're kind of routing the fluid through and then we're kind of cutting it back, and we're routing it through and we're cutting it back, or we're leaving it open because it's an especially active tissue. So there's a lot of control that we can do at these different places where we have smooth muscle, okay? Okay, so we got down to capillaries as we're doing our tour. Now we're going up the other side of the loop. So we're going capillaries into venules, which go into veins. So venules aren't really that different from veins, except they're smaller diameter. They have the same three layers, like we were talking about, the tunics, but in fact they're so small that the tunica media only has a few smooth muscle cells to it. So it's enough that they can change their diameter, but it's not a thick, robust layer like we've seen in some of those other blood vessels. And like we said, the endothelium, that inner layer, is very porous. So much like we saw with like sinusoids and stuff like that, or some of the fenestrated capillaries, we have holes which actually help for white blood cells and some other larger molecules to exit even at the venules. Because once we go from the venules into larger and larger veins, then we're going to have thick walls again. And when we have thick walls with three layers, there's very little chance that things can get across there. So it's our last chance for cells to get out, white blood cells to get out, or to do a last exchange with the tissues, okay? Okay, so we finally get up to veins. Veins are all the way around the circuit from the heart, so it's very, very low pressure, so we really need to help the blood get through these veins and get back to the heart. So what we find is that we have a thin wall to it, and we usually have a big open lumen. So if you look at the outer diameter of a very large vein and the outer diameter of a very large artery, like the aorta and the inferior vena cava, the outer diameter looks the same. But in the artery, we're going to have a really thick wall, which means that the lumen's going to be somewhat smaller. In the vein, we're going to have a thin wall, which means we have a nice open lumen. A nice open lumen means that it's easier for fluids to go through it. There's not as much resistance. Because wherever that fluid is coming into contact with the wall, it provides drag that slows it down, okay? So the smaller the lumen, the more the blood is going to be dragging along the wall. So if we want the blood to go through freely, we want a nice open lumen. And at this point with the veins, we want to do whatever we can to assist the blood to get back to the heart, Okay? Now I keep doing this because the best example is thinking about the blood coming from the lower limbs, but of course these veins are coming from everywhere in the body, routing back towards the heart. Okay, so big open lumen, a thin wall, and we said that we have these one-way valves there as well. So similar to the vessels, the lymphatic vessels that we were seeing in the lab, right? So here are the cusps of the valves. If the blood tries to come back through, it's going to catch on these cusps, and it's going to close the valve and make sure that we can't go backwards. So we're always going back towards the heart. Okay, so we tend to see blood staying within these blood vessels a little bit more because number one, the lumen's open, there's more space, and number two, there's not as much pressure pushing it through. So oftentimes veins are called blood reservoirs because if we look at our 5 liters of blood, there's a lot that is sitting in that half, if you will, of the circuit, right? In the arterial half, it's being jammed through there, pushed through there very quickly. In the capillary beds, like small brooks, if you will, like little streams, it's kind of moving through very slowly so that we can do that exchange before we then head out to the venules. And so as we're going through here, moving very slowly, low pressure, so these are blood reservoirs, if you will. There's a lot, proportionally, there's a lot of our blood that ends up in there. So about over half of the blood ends up in the veins and venules compared to the arteries and arterioles and things like that, okay? Okay, so when we were talking about arteries, we said that those are pressure reservoirs because we said they have that elastic recoil that helps to push the blood down. Instead, the veins are more blood reservoirs, that the volume of blood tends to reside in there a little bit more. So we said lower blood pressure, thinner walls, they have larger lumens, they have valves. So there's a lot of structural support or structural support of their particular function. Now sometimes we also have enlarged veins that we call venous sinuses, right? Extremely thin walls, a little bit bigger diameter where blood tends to collect. So like the coronary sinus that we see on the posterior side of the heart, where the great cardiac veins coming in, and the middle cardiac veins coming in, the small cardiac vein, that larger area on the posterior inferior side of the heart called the coronary sinus. There's also a dural sinus up on top of the brain. So definitely look at the textbook and utilize the figures that they have there. That helps you to compare these different types of blood vessels. We already did it when we were looking at different types of arteries. This is the second half, so this is table 19.1 in the new book that shows you the structure, and the structure is going to help give rise to the function, right? Thin walls for capillaries, relatively thin walls, but a little bit thicker for venules so that we can get some escape still. And then once we get to veins, we have a thicker wall, but not as thick as what we saw before in terms of arteries, okay? So being able to compare our arteries and our veins is going to be important, right? Because it's a good example of what we keep saying of structure and function. Okay, now there are some kind of smaller topics that relate to this. We talked about, for instance, vasovasorum, which is a series of blood vessels that will supply even a large blood vessel with its own little circuit, like a coronary circuit. This is a kind of another kind of smaller side note. Vascular anastomoses. Anastomoses are going to be networks that are alternate pathways that allow blood to reach a given region. So oftentimes they will occur in veins compared to arteries. But like when we're learning all the different arteries and veins on the models, not everybody's arteries and veins are going to be exactly like that. There are growth patterns that are different. So there are some people that have like 2 renal arteries on one side and only 1 on the other side, where, you know, normally anatomically we look at 1 on either side. This is a case where we'll have branching patterns that will kind of be redundant with feeding or draining a particular region. So rather than just having 1 vessel that drains the blood, 1 vein that drains the blood from that particular region, maybe there are 2 other pathways that are coming from that region. And that way, if there's a problem and we get a blockage or something like that, we've got backups, right? You've got a detour in order to be able to get around there rather than just having that tissue die off. So that happens a lot with veins, and if there are problems with veins, like last time we were talking about things like varicose veins, etc., if there are problems with veins, sometimes what they do, instead of repairing them, if they know that there are these collateral pathways, they'll just go out and remove that one or cut it or something like that and tie it off knowing that we have the rest of this network that can still handle the flow. So that's what these vascular anastomoses are. Kind of these alternate detours or additional pathways that will connect to a particular region. Okay, so to this point, what we've done is we've done our tour of the different blood vessels. We've been comparing their structure and their function, talking about what they do and what they look like. And now that we have understood the parts of the last parts of the cardiovascular system, now we kind of get into function overall in terms of, like I said, blood pressure, blood flow, and things like that. So before we do that, questions? Things we've been talking about? Yeah, Joe.
I don't understand what lipoprotein little a is. I know you went over it a little bit.
Lipoprotein little a?
Yeah.
We didn't go over that too much, really. So, in terms of when we're talking about things that are going around in the blood, there's a variety of different types of plasma proteins, there's different types of hormones and molecules that are performing a particular function in the blood, either chemical messengers or just delivering nutrients or whatever. And there are a variety of proteins in the blood that act as, for instance, escorts that are escorting, for instance, different types of fats that normally wouldn't get along with the plasma. So the plasma, right? Plasma is basically water. It's like 95% water. It's got other stuff too, other important things, but it's mostly water. So when you have lipid or fat-based substances, they don't get along. Those are hydrophobic as opposed to hydrophilic. So oftentimes they will need escorts in order to get through the cardiovascular system to get to their destination. So a lot of the proteins that we have going around can be escorting certain molecules around. So we'll get into some more detail about that when we get into digestive system as well, and we talk about the specific functions of particular factors and proteins and molecules. But at this point, we're kind of focusing on the system overall. So I wouldn't worry about it too much. Okay. So flow, pressure, and resistance. So these are all things that are going into affecting circulation. So circulation means that it's going to be moving around the loop. There are things that facilitate that help to push this blood around, and then there are things that work against it that we have to overcome, okay? So blood flow, right, the volume, a particular volume, usually like in milliliters or something like that, that's moving through a vessel at a particular time, given a particular period, often like per minute, but you could scale it up to per hour or whatever you wanted to do. So when we were talking about cardiac output, when we see CO there, right, that's not carbon monoxide, even though that's important. Cardiac output is measured in milliliters per minute. So that's looking at blood flow. Now, like this says, overall it's relatively constant when at rest, but at any given moment varies at an individual organ or tissue level based on the need. Because we have that smooth muscle where we can control the flow, just like valves on a spigot or something like that, we can increase it or decrease it. Usually we don't totally shut it off, but we can totally route it different ways to prioritize blood flow, okay? Okay, so if we go from blood flow to blood pressure instead is force per unit area. This is the push that is making the blood flow, okay? So certainly you can have the force of gravity or something like that helping to move blood, but we know that we can't just rely on that. We actually have to push it around and make sure it's going where we want to, okay? So this is often expressed in millimeters of mercury. It's always that kind of a weird unit, but once upon a time, when we were using blood pressure cuffs and stuff like that, which we will use next week, what they had is that they were connected, and if you go to an old doctor's office, sometimes they still have one like that. They have like this panel on the wall that has your units on it, so maybe it says 160 and 100 and down to 50 or something like that, right? There's units all along here, and then you can see there's this little tube in it that has this silver-looking substance in it, okay? That's mercury. So, like thermometers used to have mercury in them, stuff like that. So these blood pressure monitors, the sphygmomanometers, would have mercury in them as well. And so what would happen is you use the cuff and you hook it to the person's arm, and as the blood is pressing against the wall of the vessel, it also effectively presses against the cuff. So it exerts a pressure, an outward pressure like that. And that pressure gets kind of transferred to this, and it pushes the mercury tube around, or pushes the mercury in the tube around the tube. And so the more pressure we have pushing on it this way, it pushes the amount of mercury up and up and up, and then we read it on the meter itself. So that's why it's read, the units are millimeters of mercury. But the more pressure we're pushing on this side from your blood pressure, it pushes this mercury from this side up over to this side like that, okay? So, like we were talking about, maybe an average value is like 120 over 70. So we said that this is systolic, and this is what we're about to talk about, systolic pressure, and then this is diastolic pressure, okay? So, that's why it has millimeters of mercury, but this is the force that's causing the blood to flow at any particular instance. And blood pressure is also going to be variable. So if we change the force behind the fluid, then that's going to change the flow, so they're certainly related. So we have blood flow, we have blood pressure, and then we have resistance. Now resistance doesn't sound like a good thing, right? If somebody's trying to convince you to do something, to go somewhere, and you're resistant to the idea, you're putting the brakes on. You're like, nope, that doesn't sound good to me at all, right? So in terms of resistance, this is basically the amount of friction the blood encounters. Like we were talking about, as it's dragging along the walls of the blood vessel, it tends to slow it down, okay? So there's going to be some resistance as we're going through these different tubes, and this is called peripheral resistance, because it's out the periphery in these blood vessels that are going around the body. And there are three different sources of resistance. One is blood viscosity, the other one's blood vessel length, and the other one's vessel diameter. Now we already talked about diameter, right? Like in the veins, we want a nice open lumen, which means that most of that blood is going right through the channel and it's not in contact with the wall. So that makes it easier for it to flow through that particular area, okay? But the smaller the blood vessel, the more resistance there's going to be, right? So imagine, again, if the hallway out there, a really big hallway and you're going between your classes, there's people flowing through it, and it's not a problem. You can just go right through the center, right? But if it's a very narrow hallway and there's tons of people, then all of a sudden it's very crowded. You're bumping into other people, or you're like, whoa, I'm going to run into the wall because there's so many people there. It's going to slow you down because you have to navigate through it, right? So same thing in terms of blood. So when we have very small blood vessels, there's going to be a lot of resistance. And so we're going to have to overcome that. We're going to have to make sure that it still flows through those areas. But we know that because a lot of our blood vessels have smooth muscle to them, we can control that diameter, right? So we said the brain can control the diameter of those blood vessels. And so there are specific types of nerves or neurons that go down to them. What type of neurons or neurons are going down and telling them to constrict? So sympathetic, right? So we said autonomic is like the automatic branch of the nervous system where it's handling things at a subconscious level, right? It's like the automatic control. We said that with blood vessels, it's only sympathetic that goes down. So when we sympathetically stimulate them, they constrict, and when we stop stimulating them, they dilate, they get bigger, okay? So we can control the vessel diameter, and that's going to affect the resistance, it's going to affect blood flow, and it even affects blood pressure. Imagine, if you will, you have a hose, and it's flowing fine, and you want to stop the flow, right? You can go all the way up to the spigot and you can change the valve, but you could also just kink the hose. And when you kink the hose, that automatically is decreasing the diameter, and you can see that decreases flow downstream, but it also creates pressure upstream, right? Because you're like, and now all that fluid that wants to get through can't get through as well. So it changes the flow and it changes the pressure. So diameter of the three, diameter is the one that we actively control and the most important one, okay? Blood vessel length. To be sure, if we're talking about that hallway out there and it's really crowded and you gotta get through, if you just have to go to the room next door, it's not such a problem, right? It'll slow you down, but only a little bit because you're not going too far. Meanwhile, if it's a super long hallway and it's totally crowded and you gotta get to the other end of it, it's like traffic jam on the freeway. It's gonna take you much longer because you have to be on there a much longer period of time. So blood vessel length, depending on how far the blood is going, that's also gonna affect that resistance because it's gonna be dragging the whole way. But we can't do too much about that, right? You can't like use a sympathetic nervous system and go wink and shorten up your blood vessels and make the resistance go down. We have to make sure that they go out where they need to go. So we don't really change that, but that has a factor. Blood viscosity. When we say viscosity, what does that mean? Yeah, thickness, right? We were talking about like oil or honey or something like that. So if you are well hydrated or dehydrated or something like that, and that hematocrit, right? The hematocrit, which is the ratio of fluid to cellular content, if that is off, like you're dehydrated, you don't have enough fluid, then the blood becomes more viscous. So imagine trying to push honey through those blood vessels. It's going to be much more difficult. So if it's very viscous, that's also going to affect resistance. Okay? Okay, but out of those three, vessel diameter is the one where we tend to focus on, right? So viscosity is the stickiness of the blood. That's relatively constant because it's something we homeostatically control. If you're starting to get dehydrated, then your sweating is going to go down, your thirst center is going to get stimulated, you're going to start doing your water input, your urine output is going to go down, your digestive system is going to start trying to reabsorb or absorb as much fluid as it can. So we're trying to maintain that at some sort of homeostatic level. Same thing in terms of blood vessel length, like we said. Those are basically constant, but certainly diameter is going to be something that we change very frequently. So this resistance, when we talk about resistance and its relationship to diameter, in fact, it changes significantly, not just linear, like if I change it this much, then the resistance changes by that much. It's actually almost like an exponential change. So inversely, varies inversely with the fourth power of the vessel radius. You're like, that's a whole lot of math, I don't like that, right? But if you look at it, and you think about factor of 4, okay? So this is saying if the radius is double, the resistance is 1/16 as much. So it's not half as much. If it was linear and we doubled the radius and made it twice as big, then if it was linear, then the resistance would be only half as much, okay? But this is 1/16 as much. It's a factor of 4. So any change that you make in the diameter has a big change in terms of resistance, and that scales to flow and pressure, okay? Okay, so like we said, anything that changes blood vessel diameter is going to have a big effect on blood flow and blood pressure. So for instance, like we said, arterioles purposefully have a small diameter. They're a small vessel and they have smooth muscle that they can cinch down and protect those capillaries downstream, okay? But also anything that changes the blood vessel diameter can be a problem. So for instance, fatty plaques that can accumulate, like we talked about, that in atherosclerosis, anything that narrows the blood vessel, that's going to affect the flow downstream, and it's going to affect pressure upstream as well. And so when people have chronic high blood pressure, that can be due to problems like this, where the blood vessels are narrowed, and so that just elevates the pressure of everything. Okay, so what we find is that in terms of this blood flow, what we find is that it's going to be flowing from wherever it's high to wherever it's low. It's a bit like diffusion or osmosis or something like that. So for instance, when we are coming right out of the heart, we have the highest amount of blood pressure right there. Downstream, like a river or something like that, it's much lower pressure down there. So the water is naturally going to move from, or the blood in this case, is naturally going to move from where we have all this high pressure, where another 70 milliliters is coming right behind you. You're going to try to escape downstream and get to a lower pressure area. That's all that really means. It's not such a big deal. But if we can change the pressure and make this really high and really low, then the fluid's going to flow even faster between those two areas. Okay, so like we said, the blood pressure itself is going to be highest when we're right coming out of the heart. So the very first and biggest artery coming out of the heart is the aorta. Of course, we have pulmonary trunk as well. We have coronary arteries too, but this one's the biggest diameter. Most of the blood volume is going into that aorta. And the further it goes around this loop, the more it's going to be dragging on the walls, and the further it's going to be away from the heart. So we're going to see blood pressure the highest there in the aorta, and then it's going to be going down and down and down from there. Okay? So when we get to the right atrium, it basically is almost gone totally down to zero. And we say we're starting as something like this, right? So we're seeing this incremental decrease in blood pressure, and then that's going to affect blood flow. And so like we said, when we get around to veins, those veins need a lot of help in terms of assisting the blood to get all the way around. Now the main place where we see the blood pressure change is in the arterial. So in this graph, we're looking at all the different blood vessels there, from the point where we begin leaving the heart to the point where we enter the heart again. On the y-axis, we're looking at blood pressure in millimeters of mercury. And this graph shows us a few things. Number one, when we're looking at the blood pressure in these major arteries, we see that it goes up and goes down, it goes up and goes down, it goes up and goes down, right? It has this pulsing sensation due to the contraction of the ventricles, okay? But when we get to the arterioles here, we see a big drop in blood pressure, and we see a dampening and removal of that whole pulsing sensation. So that way, when we get to the capillaries here, when we get to the capillaries, look, it's down at like 20 millimeters of mercury or less, compared to 5 times that or 6 times that when it started out, right? So we are really trimming it down in terms of the pressure to make sure when we get to the capillaries they're protected. And it continues to drop as we go through all of these other venules and veins as well. Like I said, almost all the way down to 0 as we enter the heart again. Okay, so it's that point which is so significant at the arterioles where we're getting rid of that pulsing sensation and we're reducing the pressure so we don't blow out those capillaries. So some of these we've talked about before, right? Systolic pressure, diastolic pressure. That's the high point and the low point. So systolic pressure is when the ventricle is contracting, left ventricle. Diastolic is when the ventricle is relaxing and filling again. But luckily, we have that elastic recoil of the walls of those elastic arteries and even the muscular arteries. So those are pushing that extra volume of blood back into the artery or vein itself. So if we had an artery, when we're first sending blood down into it from the heart, what happens is it kind of balloons out like this. So rather than just going straight through like this, some of the blood goes out here into the walls or out towards and pushing the walls of that artery out, okay? So that's when the ventricles are contracting. Then when the ventricles are relaxing and filling, what happens is the walls of the arteries snap back again like this, and any of this blood that was here and here, distending the walls, is now pushed into the center channel again and pushed down like that, okay? So we still have flow, even though the ventricles aren't sending new blood down, we still have flow during this time of what's called diastolic pressure. So there's still pressure in there when the ventricles are relaxing. Okay, so systolic and diastolic are pretty straightforward, the high number and the low number, but then we also have a couple other numbers, pulse pressure and mean arterial pressure. These are a couple that we will calculate and we will measure in lab next week when we're doing our cardiophysiological experiments. So the pulse pressure is just the difference between them, right? When you're measuring pulse, that's what you're feeling. You're feeling it go out, get ballooned out, and then go back, okay? So the pulse pressure is how much pressure do we have that is causing the wall of the artery to push out like that and then snap back. How much pressure is necessary to do that? So that's called pulse pressure. And so you just subtract them. So if we had 120 over 70, what's the pulse pressure going to be? 50, right? Systolic minus diastolic, that's pulse pressure. Mean arterial pressure is not quite as easy, okay? Mean arterial pressure is just kind of looking overall. We have this high point and then it goes low, and high point and goes low. What's the mean? Now generally, if you have a high point and a low point, and you divide by 2, you get a mean and it's right in the middle. But it's not exactly that way. So you can see when we look at this pressure, see how it goes up and then it spends a longer period of time over on this side coming down again. So it's not exactly even in terms of the amount of time it spends towards the high point of the curve or the low part of the curve. So you have to do a different calculation. So for this one, it's diastolic pressure plus 1/3 of the pulse pressure. So you take diastolic, in this example 70, plus 1/3 of the pulse pressure. So 1/3 of that 50 that we were talking about before. That's not a very even number. It's a little bit easier if you have like 120 over 80. So 120 over 80 for pulse pressure equals 40, right? Because we're subtracting them. For mean arterial pressure, it's going to be diastolic, which is 80, plus 1/3 of that pulse pressure. Gosh, I thought I had a better example there. In any case, 40 divided by 3, so that's going to be about 12, 13, about 13 or so. So it'd be another 13, so it'd be about 93 for mean arterial pressure. It's not something you're going to be responsible for doing in lecture. We might ask you to do it in lab. Like if we give you these numbers, you need to tell us what systolic is and what diastolic is and be able to calculate that. But we're not going to have you do that in lecture. It's something that if you're assessing a patient, that's important because you want to know how much stress is on all of those arteries and veins, right? Because you may get them flowing out or something like that. So they do some calculations like that. So they want to monitor the pressure, they want to monitor the pulse in different areas. So that's another way that we assess the cardiophysiological health is we look at, for instance, the vital signs, right? So vital signs are going to be pulse and blood pressure, and then of course respiratory rate and body temperature. So that's often what the assistant is doing or the technician is doing right at the get-go, right? Whether you're just going to the regular doctor's office or whether you're going into the hospital, they're trying to get some of these numbers, these basic numbers. And when they're taking a pulse, number one, they have to take it on an artery because it's only arteries that are going to be doing these pulses, right? Quite often, there's a lot of different pressure points where they can do that. And so radial artery is the easiest one, right? Where the radial artery comes down right next to the radius, you can usually get access to that. And so you're temporarily compressing that so that you can feel that blood kind of come up against your finger. But there's a number of other places they can do it too. Certainly, common carotid artery. That's where you see in all the medical dramas and stuff like that. They want to see if they're dead. They're going right there. Because if you're not feeling a pulse on your common carotid artery, one of the biggest ones right next to the heart, there's not a lot going on. So you could have low blood pressure and a very, very low kind of metabolism going on and not feel a good pulse out at the periphery because it's very weak. So you might check up here where it's very close to the heart and see. So common carotid, but there are other places, brachial, femoral, etc. This one down here, dorsalis pedis. So the dorsalis pedis is an artery that comes down here and then it goes into a loop on the dorsal side of the foot. Dorsalis pedis is one that they often check at the hospital because you might have IVs here, you got your tags there and all this stuff. So maybe this isn't very accessible or maybe it'd be uncomfortable to be checking that regularly. So feet, usually unencumbered, so you can check that one instead, okay? But you should know at least a couple of different places where you could check a pulse, where you could palpate, right, feel a pulse. Okay, so we talked about blood pressure, I'm pushing these things around. We talked about how arteries pulse and things like that and how veins don't. This is one of the other aspects of the cardiovascular system that helps the blood to go around. Not only do we have pressure from the ventricles pushing it around, but we said we have assistance in other ways too. Number one, we know that veins have valves in them, like we see here, so that assists them in moving it along. We know that the veins have smooth muscle in their wall, so they can kind of squeeze themselves and help to squeeze it along. But also, often veins go through these different areas of skeletal muscle. And so like we were talking about before, when the skeletal muscle contracts, it tends to squeeze the veins that are going through them. And so by doing that, again, it's like squeezing the toothpaste tube and moving the fluid through. And with these valves, it doesn't matter where along the tube you squeeze, the blood can only go one way. It can only, in this example, go to the left. So it'll go through the one-way valve. If you try to squeeze it and it runs back the other way, the valve closes. Okay? So that assists in blood flow as well. It's called the skeletal muscle pump. And that is squeezing all of the veins and helping that blood to return. There's also what's called a respiratory pump, but this one doesn't contribute too much, okay? The fact that the heart is located in the same cavity, thoracic cavity, as the lungs, when we talk about the respiratory system, most of the time what we find is that there is kind of a low pressure in the lungs which aids in us drawing in air. And so what we find is the changes in pressure that are created by ventilating the lungs creates a lot of times a lower pressure in the thoracic cavity. The heart's in that cavity, so it kind of benefits from a slight lower pressure where it's located in the body. So if you were like laying down or something like that, you would get a little bit of extra draw back towards the thoracic cavity because this is kind of a center of low pressure. So it's a slight pressure that helps to draw blood back to the heart, but it's much lower than a lot of these other types of factors that we're talking about, but it does contribute. Okay, so what we've seen is we started talking about the heart, we then talked about the blood vessels, and when we think about it, right, it's all part of one circuit. And so there are changes or activities that can happen in either place on the circuit that then affects blood flow. So if we have changes in, for instance, cardiac output, the number of squeezes per minute or how strongly we're squeezing each time, that's going to affect blood flow because that's the 70 milliliters that's getting pushed out each time. But also, if there are changes in the vascular part of the cardiovascular system, like changes in diameter of the blood vessels or whatever, that's going to affect resistance. That's going to affect the ability of the blood to flow through. And then we said blood volume and blood viscosity. So all of those things will contribute to blood flow and blood pressure, right? Factors associated with the heart, factors associated with the vessels, and factors associated with the blood itself. So all those things come into play. So if there's a problem with blood pressure, like it's chronically too high or something like that, they start looking at, okay, what can we fix? What's easiest to fix? Number one, what's the root of the problem? And unfortunately, with a lot of hypertension, that's what they call hypertension, chronic high blood pressure, with a lot of hypertension, they don't know exactly what the problem is. It's too kind of diffuse to figure out what it is. So what they tend to do, the easiest fix for decreasing hypertension or decreasing blood pressure, is they tweak blood volume. So what they do is they'll put somebody on a diuretic, and what does a diuretic do? Diuretic makes you pee, right? So like caffeine and alcohol and things like that, those are diuretics, and so you end up losing more fluid than you're gaining. Now in terms of the cardiovascular system, if you have too much pressure, if we reduce the amount of fluid that's in all these arteries and veins, then necessarily the pressure goes down. So if we get rid of a little bit of that fluid, and we have less than 5 liters or less than we normally do, then the pressure automatically goes down. It doesn't solve the core of the problem, the root of the problem, but it still decreases the pressure overall. So it's kind of a quick fix to chronic high blood pressure, is to change the blood volume instead. Okay, so that's an external fix. If a doctor sees a problem and wants to fix it, then that's one of the things that they can do. It's kind of an easy way to do it. But what are the controls that we have inside of the body where we're constantly using homeostasis, we're constantly monitoring and adjusting and comparing it to the set point? So how does that work? We know that it's always going to be like a negative feedback loop. We're going to have sensors, we're going to have a control center, we're going to have a set point, and then we're going to have effectors where we can affect or produce an effect or cause a change to occur. So we've got lots of different ways that we can affect control blood pressure, to increase it, to decrease it, to make it exactly what we need to. And that's not only overall blood pressure, like this type of thing, but blood pressure and blood flow to particular areas, right? We can have an increase in blood pressure in particular areas of the body, which is totally different than the blood pressure and blood flow in that other area. Like you said, going to the stomach and the intestine rather than the muscles or to the brain or something like that, okay? So what we find is that there are some short-term controls as well as long-term controls, and some of them are going to be nervous, some of them are going to be like hormonal, and some of them are actually local controls, little chemicals that are released from a local area which says, in that area right now, I need some more blood flow. Like for instance, if I have a cut on my forearm right here, we know there's all kinds of things that are going on specifically right there. We have a clotting cascade going on, we're going to be plugging up that blood vessel. Uh-oh, we got to rely on other blood vessels now, so we have a change in blood flow. But in that particular area, we want to plug up that vessel, but we also want to make sure that the area around where that cut is still has blood flow, so we can maybe take white blood cells there, because bacteria might be getting in there, so that we can increase oxygen and nutrient delivery to the area, because those epithelial cells of the skin that were damaged or removed, now we got to go through a whole bunch of mitosis to replace them. So you get kind of that swelling and redness around a cut as well. So there's localized changes that can occur even on the order of like a centimeter or something like that. Like this area right here might be red because we have increased blood flow right there. If you hurt it or if there's something that needs an increase in flow. Okay, so what we see is that there's lots of different ways that we can control blood pressure and blood flow. So a couple of short-term ones in terms of regulation or control, neural and hormonal. That's always going to be the case. Almost every system that we get to, as we get towards the end of the system, it's like, okay, how does this work with other systems? How do we make sure that it's giving us what we need at different times and different requirements is we're going to have a wired control, neural, from the brain and spinal cord. We're going to have hormonal control from hormones from different types of endocrine glands. And then, oftentimes we said we have local control as well. Now, in this case with blood pressure, we actually have another organ, an organ system that helps, and that's called a renal control. And renal, you guys know renal artery and renal vein, where's that going?
Kidneys.
Kidneys. So this isn't just saying that the kidneys like get bigger and get smaller or something like that as they swell with blood. What it means is that kidneys, their main job, as we'll talk about later in the semester, is to help control blood volume and blood concentration. Their job is to filter the blood and make sure that that concentration and that balance between cellular elements and fluid components is about right. So they have a very high flow rate and they're always checking it. And if it gets off, then that's one of the places that we feed back and say, okay, we need to make some adjustments. We saw that already when we were talking about red blood cells, right? Erythropoietin, which causes an increase in red blood cell production from where? From the red bone marrow, right? That's from the kidneys. The kidneys are looking at this stuff all the time. So there are also some renal controls, some long-term controls that are coming from the kidney itself, okay? Okay, so if we look at some of these neural short-term controls, what we find is, okay, where in the brain are we helping to control, or what aspects of the brain and the nervous system are we using to help control blood flow and blood pressure, etc.? So, one of the main places of the brain is the medulla. Now this is actually the medulla oblongata. I have no idea. It's so interesting that, to me, right, we're learning all these specific terms for all the different parts, etc. And I don't know about this trend recently where the medulla oblongata, they've been kind of truncating it to medulla. That doesn't make sense to me at all. Because like we were just talking about, or like we will talk about, like there's lots of things that have a medulla, an inner core to them. So if you just reduce that to medulla, you're like, now it gets very ambiguous. So we're talking about the medulla oblongata, the lowest part of the brainstem that you guys learned before, right? So you got the midbrain, you got that fat-looking pond sticking out on the anterior side, and then you have the medulla oblongata before it transitions to the spinal cord. The lower areas of the brain are what are handling life-sustaining functions. It's not where you're dreaming and getting creative and poetic and stuff like that. That's way up here. Base of the brain and the brainstem, it's all about keeping you alive. So it makes sense. Cardiovascular function, that's as low as you can get. We want to make sure that that is always occurring. So, vasomotor center is going to be in the medulla oblongata, and in terms of, for blood vessels and stuff like that, right, that's where we're going to have a lot of sympathetic neurons coming out. There's also a cardiovascular control center that's located in the medulla oblongata that not only has neurons going to the blood vessels, but it also goes to the heart. So remember, sympathetic and parasympathetic at that point to increase its heart rate or decrease the heart rate accordingly. So we mess with the heart, we mess with blood pressure and blood flow. We mess with the blood vessels, that also messes with blood pressure and blood flow. Okay, so like we said, sympathetic activity, we're going to have vasoconstriction, and if we constrict it, the diameter is going to get smaller, and that's going to increase resistance, okay? So if we constrict it, what's going to happen is we're going to get generally an increase in blood pressure because we're creating a backup in those blood vessels that are more narrow now, okay? So, in terms of, that's going to be the control center in the cardiovascular center in the medulla oblongata. But the question is, how does it know what's going on, right? It wants to maintain it at 120/70 or something like that, but how does it know what the current situation is? It has to have receptors to tell it. So we have certain types of receptors, and these are pressure receptors, baroreceptors, right? So a barometer, right, these days with storm systems coming in, stuff like that, if you hear about the weather, they're talking about, oh, it's a high pressure system, it's gonna be hot and clear, it's a low pressure system associated with a hurricane or something like that, and so now we have instability, blah, 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 we may even get some rain, who knows, right? So that's from a barometer, measures atmospheric pressure. These are baroreceptors which measure blood pressure in distinct areas around the body. So what we find is baroreceptors are going to measure pressure. There are some that are in the carotid arteries. That's going to be important. Why? Where's that blood going? brain. We want to make sure there's enough blood pressure and blood flow going up to the brain at all times. There's also some baroreceptors in the aorta. Where's the blood in the aorta going? Everywhere else. So that's a good checkpoint. We want to make sure that it's good there too. Okay? So, different types of baroreceptors in different places monitoring the pressure. Chemoreceptors. So, chemoreceptors are measuring chemicals, right? And in this case, we're looking at some of the different chemicals that are going to be found in the blood. So, not only do we want the flow to be good, but we want to make sure that whatever is getting to that site has what it needs. So if, for instance, the oxygen levels are getting low, I don't care what the pressure is, we need to push some more over there because that tissue is not getting enough of this critical molecule, okay? So we need to make sure that the oxygen levels are staying high enough, that the carbon dioxide levels aren't too high. We want to make sure that those are low. So we want good flow in there to circulate around, deliver oxygen, remove carbon dioxide. If those are getting out of whack, we can change the blood flow to kind of flush that out or deliver more, okay? Hydrogen ions, what do hydrogen ions generally have to do with? pH, right? Has to do with whether acidic or basic it is. That's important. We're always maintaining a certain pH in the body as well. Specifically, this will relate to this when we talk the respiratory system. The more carbon dioxide you have in the blood and the tissues, the more acidic they are. The more active your tissues are, right? You're talking about muscles and lactic acid buildup and stuff like that, anaerobic respiration. If they're really active, they get more acidic. That's a good thing to monitor to say, ooh, that tissue is really active. That probably means it needs more oxygen. That means it's going to be producing more carbon dioxide, so we need more blood flow going through there. Okay? So we have chemoreceptors that are monitoring things like that. Now, we also will have information coming in from these receptors, and then, of course, there's going to be a variety of other things that can affect our vasomotor activity too. Higher brain centers. So what that means is, right, higher brain centers physically, we're talking about higher, kind of, but we're talking about like higher thought, so cerebrum, etc. So right now, if you have a certain blood flow that's going on, maybe it's kind of low, you've been sitting here for a while, so you're slowing down and your blood pressure is getting lower, etc. But if I start reminding you that we have an exam 1 week from today, and you think you have plans this weekend, and you better cancel them because you want to do good on this first exam, then all of a sudden you're like, oh crap, I got to reschedule this, I got to see if I can get out of work, I got no time to study, I got this I planned, I forgot about that exam next week. I guarantee you that your blood pressure is just now increased. And that has to do with not demands more in the body, you're not getting up and running or anything, those are emotional stress that's going on. So higher control centers, right? Higher brain centers also affect blood pressure, okay? So there's some influence of higher brain centers as well. Bloodborne chemicals, there are some chemicals that are circulating around that can increase or decrease flow. We talked about that in terms of like local blood flow, etc. And hormones. So hormones, think about like adrenaline, right? If you were driving to school here and somebody almost ran you off the road, you know your heart rate starts going and it stays up for like an hour or something like that, and then it starts slowing down, right? That has to do with epinephrine, norepinephrine flowing around and then affecting heart rate and vessel diameter and things like that, okay? So normally what's going on here is we're trying to maintain balance or homeostasis. And if something upsets that, like the blood pressure goes up, like we were talking about, oh, an exam next week or something like that, then what happens is we're going to have these receptors which tell the brain that pressure has just gone up. Now, pressure has just gone up, we have to see, okay, is there a need for that? Why did that just happen? And so we have the sensory information that comes back to the brain. The brain in the control center, specifically like down the medulla here, is saying, okay, do we need it to be up? Is that important? Can we maintain that? Do we need to maintain that? Or do we need to kind of counterbalance it? So what we can do is, if blood pressure just went up, what we want to do is we want to counteract that. So we're going to decrease the amount of sympathetic impulses to the heart and to the blood vessels. And that is going to decrease cardiac output. It's going to dilate those blood vessels and decrease our resistance, so it makes it a little bit easier for the blood flow. And what that's going to do is it's going to decrease our blood pressure. So what we find is blood pressure is going to go down again, and we've gone back from being out of whack back to some homeostatic set point, okay? But we're constantly going to be monitoring that and adjusting it all the time, whether we are decreasing, like we see here, or maybe increasing as well, right? If blood pressure goes too low, like we see here, then we're going to need to bump it up again. It's just like the thermostat in your house. Constantly monitoring. If it gets a little bit out of whack, we nudge it back, one way or the other, okay? Okay, so neural controls, sympathetic mainly, some parasympathetic to be sure. Chemical controls, we said oxygen and carbon dioxide are going to be important. We have chemoreceptors that are in those same places where we tend to find our blood pressure receptors, those key places where the blood's going to be going. We want one last check before we send it out into the world, into the body, etc. So in these cases, we're monitoring those levels and make sure that we have enough going to the body and to the brain, etc. So we'll have, what they tend to call them is, there are both central and peripheral receptors. Central ones will actually monitor what's going on in the brain. Do we have enough oxygen in the brain? Is it too acidic in the brain and the spinal cord, etc.? And then we'll have additional ones that are out at the body outside of that too. Okay? Okay. Chemicals that increase blood pressure. These are some examples of chemicals that can increase blood pressure. We talked about these two, right? That's adrenaline and noradrenaline. What you just experienced when we started talking about the exam 1 week from today. Oh, are we getting that? Okay. So, we have hormones that will affect it in terms of directly affect blood pressure, heart rate, and blood vessels and things like that. But we also have other ones that can affect blood pressure. This one's called antidiuretic hormone. So if we look at that one, what did we say a diuretic does? Makes you pee. So this one effectively is no pee hormone. Okay? So what that does, that's released by the pituitary gland, and it has a few different effects around the body. Number one, it causes you to reabsorb water in the kidneys so that less is lost as pee. So it's like, blood pressure is getting too low, so we want to hold on to whatever we have. So antidiuretic hormone is going to cause you to hold on to fluid, which makes the blood volume go up, which helps to maintain blood pressure. It also can act as a vasoconstrictor. So if, again, blood pressure is getting too low, if we constrict all these blood vessels, now that blood is at least contained within a smaller area, so it maintains the pressure. Okay? Angiotensin II. Angiotensin II is part of what's called the RAS mechanism. We'll get into this a little bit later. So renin that we see here comes from the kidneys. This is where the kidneys are monitoring everything. It activates angiotensin II, which activates a hormone that's called aldosterone. And this steroid hormone called aldosterone, we're gonna have a screen. Aldosterone has an effect on its own, but angiotensin II causes vasoconstriction. That's going to help increase blood pressure. Aldosterone causes us to hold on to sodium. Now, you know, if like this weekend where you're not having any fun, cancel those plans, and you're sitting at home trying to stay awake, and you have this big box of Cheez-Its that you're mowing while you're trying to stay awake. I do the same thing when I'm trying to grade, right? Just have the box right there. And I'm not even gonna kid myself. Oh, why put it in a bowl? I just go straight to the box, right? But these are low fat, so I can have twice as many, right? Lots of salt in that. Salt makes you feel bloated because salt creates an osmotic gradient. So you're piling in all the salt, the water tends to follow the salt, and now we're increasing blood pressure. So on a certain level, the motion helps to stay awake, but it also works to increase your blood pressure, which increases blood flow, which hopefully helps you to keep awake as well. But aldosterone as a hormone also has that effect. So if that hormone gets released because blood pressure is getting too low, it starts to hold on to sodium, and then water is going to follow the sodium, and that's going to increase blood volume. Okay? These ones here, the endothelium-derived factors, those ones, as well as these other ones here, don't worry about the whole acronym and stuff like that. But these ones are basically little local chemicals that are produced when you need to change the blood flow in an area of the skin or the particular tissue, etc. So there are things that are produced by the tissue that can cause vasoconstriction and also vasodilation to, like we said, increase the blood flow to a particular area. So for instance, if I do this across my forearm, right then when I was doing it, it turned white because I was actually physically driving the fluid away, right? But if we give that a couple minutes or so, you're going to see that there's going to be a red mark there. So what it's saying is, oh, there is, a little bit of stress there on the tissues. We better send some of our troops over there to make sure everything's okay, that we didn't damage cells or remove them, bacteria aren't getting in there, etc. So I didn't do anything except for draw my fingernail across my arm, right? But I changed the blood flow right in that area where I just did that. Okay? So there has to be localized chemicals that can say, whoa, something happened right here, we're going to need some help. We're going to need some increased blood flow for a particular time. Okay? Okay. So, now when we were looking at this, these were all chemicals that would increase blood pressure. Blood volume or blood pressure was getting too low, we needed to jack it back up. We need to push it back up in some particular way. These ones are working the opposite side of the coin, decreasing blood pressure. Again, like the autonomic nervous system with sympathetic and parasympathetic, we like to have something that works antagonistically, something that works like a gas pedal and something that works like a brake. Okay? So we have chemicals that can work to push it up, but we also have chemicals that can work to push it down. So this one's an interesting one. Atrial natriuretic peptide hormone. Okay? So when we look at this one, this is peptide based, it's like a small protein, okay, that just says the chemical nature of it. Atrial, what does that remind you of? Atrium, okay? So what happens here is if blood pressure is too high, then the atria are where all this blood is piling back into in order to go back out again. So if the blood pressure and the blood volume is very high, then those atria are going, whoa, that's a whole bunch that I just got back in preload, and they're getting really stretched. And that's too much stress on them. And so if they get too stressed like that because there's that much blood coming back or too high a pressure, they're going to send a message out. That is too much. We need to tamp that back down again. So they actually release this peptide-based hormone called AMP, and what it does is it helps to decrease the blood volume because when you look at this, Na, if we look at like a chemical chart, right? So we looked at the periodic table. So what is Na? Sodium. It's sodium. Okay, we know that sodium affects water, and so the effect of AMP is that at the kidneys we start getting rid of that sodium, and if we get rid of sodium, the water is drawn out with it. When we lose water, that's going to decrease blood volume. It decreases blood pressure, and now that's easier on those atria. Okay, so AMP, this one, kind of works opposite to these ones that we see here in terms of epinephrine, antidiuretic hormone, and even the RAS mechanism like we were talking about. These ones here are some that, again, are released by local tissues. Nitric oxide is found all over the body as a chemical messenger, a local chemical messenger, so it can be a vasodilator. And then there's different types of inflammatory chemicals. Histamine, we talked about that one already, right? There's a white blood cell that releases that. What's that one? So what we have is different types of white blood cells. So we have our eosinophils and our basophils that are associated with our different types of inflammation and things like that, right? So histamine is going to be coming out of there, but what we also find is that there are other types of localized chemicals that act as vasodilators to increase blood flow as well. See? Increased blood flow right there on my arm. Okay? So that's what happened. We had some inflammatory chemicals coming out. This one, this one is an extrinsic one, right? So this one, you should not be utilizing this weekend. So this one can cause a blood pressure drop by inhibiting antidiuretic hormone, right? So it is a diuretic. The main way that it's a diuretic is it keeps us from releasing antidiuretic hormone. Antidiuretic hormone says, okay, we're going to hold on to water down the kidneys, we're going to constrict blood vessels, etc. Alcohol actually inhibits that whole pathway. So if we're not doing that, that means that in the last tubular segments of the kidney, it's just shunting straight through. So we actually end up losing, I get it, right? You're like, no, I'm hydrating. Look, I'm drinking the whole weekend. I'm drinking, right? Even though you are taking in a volume, you're losing a larger volume out the kidneys because you don't have that antidiuretic hormone trying to hold on to it. So through time, you end up being dehydrated because you're losing that extra volume. Okay? So this is going to overall can act as something that decreases blood pressure. You know, so that passing out thing comes from all kinds of different reasons. Okay? Okay, so we do have a little bit more to go in terms of different types of control and things like that, but we will work on that, finishing up the chapter on Tuesday of next week instead.